Hello students, welcome to our next life science lesson. Today we will be looking at DNA profiling, and what you would be required to know by the end of the lesson is the definition of a DNA profile, the uses of DNA profiling, and you need to be able to interpret a DNA profile. So what is a DNA profile? Here's an example of one. If you look at it, what you can see is a pattern of black bars that represent DNA fragments. And if you recall that every individual's DNA is unique, that means that every person's DNA profile is going to be unique as well. If we look at some of the uses of DNA profiling, I'm sure you can give me the first one by actually looking at this particular DNA profile. What do you think it is? DNA profiling can be used to identify criminals. It can also be used to identify relatives. It can be used to identify paternity, and it can be used to identify dead bodies. In addition, your DNA profiling can be used to diagnose inherited genetic disorders in the unborn and in newborn babies. So if we look at that again, the uses of your DNA profiling, it's to identify individuals, whether the individual is a criminal, a relative, it's about paternity and dead bodies. What do you mean by paternity? Paternity is about who is an individual's biological father. And the last use that we mention is to diagnose inherited genetic disorders. Let's look at some sources of the DNA, because if you are to make a DNA profile, it means we needed to extract the DNA from a cell in the first place. So where do we get these cells from? If we look at our next diagram, we can see, for example, blood. Blood is an example of a tissue. So one place or one source of your DNA for profiling are tissues or cells of the body. The other would be body fluids, and the body fluids would include blood, saliva, and semen. You also find traces of DNA in sweat, urine as well, but they don't yield as much sample as the blood, semen, or the saliva. We also can get DNA. If you look at our diagram, you may see a hair follicle. Now just remember here, if you are using a strand of hair as the source of DNA, it's got to be with the root, because your strand of hair is merely a protein, that's keratin, and that doesn't contain DNA. So your DNA would be in the root. So let's think about that crime scene again and how these sources may apply. In a crime, let us say it was a murder, there might have been a struggle between the victim and the perpetrator of that crime. In that struggle, the victim may have scratched the perpetrator. If that's the case, then under the fingernails you would find skin cells of the perpetrator. You may have seen this on television dramas where they talk about forensics, where they do scrapes under the fingernail. That's an example of where that would apply. If we look at blood as an example, in a violent crime, blood is going to be spilt, and it may not only be the victim's blood. If there was a struggle, there could be the perpetrator's blood as well. Let's look at saliva. In the case of a victim knowing the perpetrator, perhaps they had drinks together and glasses are left at the scene of the crime. By analyzing the saliva on the glasses, you may find cells in it. Your cheek cells are very loosely bound, so they are often being shed and they are in that saliva as well. If we look at semen as being a source, obviously in cases of rape, the rapist's semen would be found in the victim. You may notice some other diagrams that we have. There's teeth and bone. So these can be sources as well. But again, they may not yield as good material as you would from the other sources we've mentioned. In fact, another source could be feces. Often when we study animals, you find that you can detect changes in a population by looking at the DNA from samples of feces. Now, how do we obtain our DNA? How do we get it from being in some cell to being an image that we looked at, right? So let's look at our next diagram here that shows the process. So if you have a DNA sample, firstly, you use chemicals to extract the DNA from the cells. Once you've extracted the DNA is then cut, and it's cut using enzymes. We call them restriction enzymes. They cut particular fragments of DNA that scientists know are markers to distinguish one individual from another. These fragments of DNA then undergo a process called gel electrophoresis. What this process does is it separates the longer fragments of DNA from the shorter fragments. This is then placed on a membrane, and radioactive probes are then added to the DNA on that membrane. Radioactive because radioactive materials show up on X-ray. So once your radioactive probes are added, an X-ray is done, and if you look at that image, you can see that distinctive image like a barcode. That is your pattern of your DNA profile. What's very important for you to be able to do is interpret a DNA profile. These are very popular in the exam questions that you would get. So how would you interpret a DNA profile? And let's go back to the first profile that we looked at. We can actually see three profiles on the one side, and there's one profile. So if we look at this particular profile, we can see we've got four profiles. The first one says crime scene. The second one says suspect 1. Then you've got suspect 2, and then you've got suspect 3. Now, crime scene means that that particular DNA profile was taken from a sample found at the crime scene. So maybe it was a blood sample that was taken, the DNA extracted, and the profile made. And then in crimes, we always get suspects. So 3 suspects, their DNA profile was taken. And how is that often done? So if somebody is a suspect in a crime, to get a DNA sample from them, you do a simple cheek swab. So by doing a cheek swab, you get the cells that are loosely bound in your cheeks, and you extract your DNA from there, you obtain your profile. Now, in interpreting, particularly in cases like this where it's a crime, what are we looking for? Notice the patterns of the black bars on your DNA profiles. You find what we look for are matching bars. So, if the black pattern on a DNA profile in one sample matches the black pattern on the DNA profile of another sample, we will assume that the 2 samples came from the same individual. That's the way we do it. So we look at these matching black bars. And to be sure they came from the same individual, it can't just be 1 or 2 bars that match. Because amongst individuals, you will find some matching bars amongst different individuals. But in order for it to be a match, we look for a perfect match. Every bar has to match. So you look at that carefully. What can you see? Which individual, which suspect matches the sample from the crime scene? I'm sure you can easily pick up that suspect 2. You will notice if we look at the first bar at the crime scene, it matches suspect 2. If you look at the second bar, it will match suspect 1 and 2. So as we said, some bars from different individuals may match. But if you look at suspect 2, every bar matches the black bars on the DNA profile found at the crime scene. Okay, so remember, that's a key thing, the matching of the black bars on your DNA profile. Now, in many cases, and you may have heard of them, right, in rape cases, in paternity cases, in murder, that DNA profile has often been conclusive evidence because we know the DNA of an individual is unique. So why is it that sometimes in a criminal case that even though a DNA sample particular individual, that individual may not be convicted of the crime? Why? Well, one is that an individual could have been at a crime scene before the crime was committed. It could be someone you know, so a family member, a partner, a friend could have been at the crime scene before the crime actually happened, and their DNA evidence may be in a particular space. Another is there could be human error in obtaining the DNA profile. For example, in a laboratory, it's possible that there was contamination of a sample. It's possible that the analysis wasn't accurate. It's possible that some inaccuracies happened in the technique. So human error is another issue. Then we come to ideas like evidence being planted at a crime scene. We know it's possible. Let's just say someone is a well-known criminal, and you make it a scenario where the police think, we know he's committing all these crimes, or she, and we can't get the evidence to convict the person. So out of frustration, we know it sometimes happens, where evidence can be planted at a crime scene to convict a particular individual. That's where there's a dishonest practice. So that's a possibility. Another is, although this seems an unlikely scenario, that someone may have an identical twin who's a criminal. We know individuals have different DNA except for identical twins, where they've got the same DNA.
We need to learn to live with the COVID virus, you know, the new normal, at least for the next few months. And we have just what you need to make sure you never miss a single school lesson. And it's absolutely free, mahala! High quality, powerful, animated lessons for all your subjects. So don't spend a cent until you have tried IWAS. It's everything you need. All video lessons, textbooks, workbooks, exam prep. We want you to ace 2021.